Today we'll be talking about Nirenberg's and Matthei's and Nirenberg's and Leder's experiments that led to the discoveries of genetic code. During this time, it was understood that DNA directs the synthesis of RNA, and in turn RNA acts as the template in protein synthesis. However, scientists did not understand how the genetic code was translated. So Nirenberg and Mattheis were the first to determine that a specific RNA sequence coded for a specific amino acid. They did this by first preparing an extract of E. coli cells, which contained cytoplasm and ribosomes. They would then add a series of 20 amino acids, one of which would be radioactive, and a homopolymer, in this case UUU, to the extract. The radioactive amino acid is able to make proteins glow, allowing them to track which amino acids are making which protein. The scientists found that the UUU homopolymer extracts produce a certain protein, phenylalanine. And this procedure was repeated 20 times, with each tube containing a different labeled amino acid. They ultimately found that the test tube in which the protein was radioactively labeled contained newly synthesized protein with the amino acids specified by the homopolymer. So at first, this could only be done to homopolymers such as UUU, AAA, and CCC. So a few years later, Nirenberg and Leder were able to determine genetic code for all codons, not just homopolymers. Their procedure was nearly the same as their last experiment. So they started off by synthesizing artificial mRNA codons and adding it to a mixture of ribosomes and tRNAs attached to amino acids, one of which is radioactive. Some of the tRNAs ended up binding with ribosomes and mRNA, while some do not. The mixture was then passed through a nitrocellulose filter that traps the tRNAs paired with ribosome-bound mRNA at the top, letting unbounded tRNAs through. Essentially, this allowed for all radioactive proteins to stay at the top, allowing them to determine which amino acid sequence was being translated into a protein. For example, when mRNA with GUU was added, the tRNAs on the filter were bound to valine, and therefore the codon GUU specifies for valine. They were able to identify 61 codons using this method, which is this genetic code table right here.